Hello everyone, this is Soumya, working as an assistant professor, Department of Zoology, Government First Grade College, Vijayanagar, Bangalore. Today, I'm going to explain few things related to ABO blood groups in humans. Before getting into the main topic, let me say few things regarding the blood. Blood is a mobile connective tissue which is there in the animal's body. Blood comprises of the plasma and cells. Blood is the basis of life and it is the softest tissue in the body. The fluid which is there outside the body or the cells will call that as extracellular fluids. So in that blood composed of 30 to 35% of extracellular fluid. So the volume of the blood, usually if a person who's weighing around 70 kgs of weight will be having 5.5 liters of blood. And the blood is slightly alkaline in its pH. pH, you all know that the acidic and the basic condition in the liquid decides the pH of an organism or in the pH in the media. In case of the human, the neutral pH is the 7, whereas the pH in case of the blood is slightly alkaline, that is 7.4. And the pH in the arterial blood is more than in the venous blood. Arterial and the venous blood is like, you all know the circulatory system in case of the human. It is a closed type circulatory system which is having arteries and veins. Arteries are the one which are carrying the oxygenated blood. In turn, it is called as a pure blood. And the veins are the one which are carrying the deoxygenated blood. In turn, the impure blood. You can notice the blue color veins in your wrist. So those are the veins which are actually carrying the deoxygenated blood. That's why it appears blue in their color. And as I said, the composition of the blood, it comprises of the plasma and the cells. So the plasma is slightly alkaline, which is 55% of the blood is composed of plasma. When it comes to the color of the plasma, it is yellow, transparent, and clear fluid. Plasma comprises the various components. These are the components which are there in the plasma. We'll discuss them in detail. Water, as we all know that 80% of the organism body is composed of water. So in that, plasma comprises of 90 to 95% of water in its composition, and rest 88% is the solids. And when it comes to the minerals, some vital necessary minerals are also there in the plasma, like the sodium, calcium, magnesium, bicarbonate, chloride. These kinds of vital minerals are there in the plasma. When it comes to the nutrient part, as the blood is the circulatory fluid throughout the body, the nutrient is also there in the blood components. So like the nutrients like the glucose, amino acids, lipids are there in the blood. When it comes to the protein, various proteins, about 6 to 8% of proteins are there in the plasma. Like proteins like the albumin, globulin, and the fibrinogen proteins are there in the plasma. And defense components. Defense components are nothing but the components which are involving in the defense mechanism. Defense mechanism is otherwise the immunological mechanism. Here we can see the immunoglobulins and the lysozymes, a polysaccharide, and the propyridine, which is a protein, large protein. These are the components which act as a defensive components in the human body, and they are also there in the plasma. And it also having the excretory substances. As I said, blood is a liquid media where that will be circulating throughout the body, and all our cells will be involving in the metabolic processes. At the end of the metabolic process, there will be the release of excretory substances like ammonia, uric acid, urea, creatinine, those kinds of excretory products which will be released into the blood. So the excretory products will also be there in the plasma. And the dissolved gases. The major role of the blood is to transport the gases. The gases, dissolved gases like the oxygen and the carbon dioxide, even the nitrogen will be there in the plasma as dissolved gases. Anticoagulant. So anticoagulant plays a very major role in maintaining the consistency of blood. We all know that blood is liquid in nature. and that consistency has to be maintained if at all, and that is maintained by the anticoagulant. So it maintains or prevents the coagulation. Blood is composed of a components which are proteinaceous in nature. They may undergo coagulation and block the arteries, so that will be prevented by the anticoagulant, which is a heparin, a conjugated polysaccharide. If at all, a heparin, the anticoagulant, if at all it is not there in the blood will get thickened, that will get coagulated and blocks the arteries, that even cause the fatal to the organism. And the hormones. When it comes to the hormones, we know that the endocrine and the exocrine hormones are there. Endocrine hormones are the one which will releases their secretion directly into the bloodstream. So those hormones will be circulating in the plasma, in turn the blood. And vitamins and enzymes, various vitamins and enzymes which are needed by the cells for the various purposes will also be circulating in the plasma. And factors for clotting of blood. So I said anticoagulant is there to maintain the blood clotting or the coagulation inside the arteries and veins, whereas the clotting factor, these are there in order to prevent the blood loss when the mishappenings like some wounds or the incision happens to organism. So the clotting factor which are there in the plasma takes the role to prevent the further oozing or the loss of the blood. When it comes to the functions of the plasma proteins are involving in various functions like prevention of blood loss. As I said, in the mishappenings, the clotting factor takes a role in preventing the blood oozing and the bleeding by clotting the wound. Like the fibrinogen and the prothrombins are the proteins which are there in the plasma, helpful for the clotting of the wounds and retention of fluid in the blood. The components like albumin and the globulin takes a major role in retaining the fluid content in the blood. And the body immunity. So the immunoglobulins which are there in the plasma, they'll be helpful to prevent the infection of various foreign components by killing them with the immunological reactions. And maintenance of pH. As I said, the neutral pH in case of the human body is 7, and that will be maintained by the components which are the proteins which are there in the plasma. The plasma protein also helps to maintain the pH by forming the acid base buffer so that it maintains the neutral pH of 7. And transport of certain materials. As I said, there will be the transportation of the endocrine hormones to the different organs. I mean, the cells of need, the cells which are needed of those hormones will be transported through the plasma. For this, I can quote an example like the thyroxine hormone. The thyroid gland, which is there in the neck region, it releases the thyroxine hormone that will bound to the albumin, which is there in the plasma, and reaches the cell where the thyroxine is needed. So this is how the transportation of certain materials occurs in the plasma. And the distribution of heat. So you all know that we humans are homeotherms. Homeotherms are the one which maintain the constant body temperature of 37 degrees centigrade. And in order to maintain the constant body temperature, we are provided with a mechanism. So we are the endotherms which we can create the heat within us. So that distribution of heat occurs with the help of the plasma protein. So they'll take part in uniformly distributing the heat or the temperature so that the organism can remain with a good temperature, and that also helpful to maintain the physiological processes. And enzymes, various enzymes which are proteinaceous in nature or also be there in the plasma. And as I said, the blood is composed of plasma and the blood corpuscles or the blood cells. In that, the first ones are the red blood corpuscles or the erythrocytes. So when it comes to the number of the erythrocytes, so the normal man is having 5 million RBCs per cubic millimeter of blood, and the normal women will be having 4.5 million RBCs per cubic millimeter of blood. And the less in the number of RBCs leads to a condition, what we all know that the anemia is a condition where there will be less number of RBCs, and the abnormal rise in the RBCs leads to a condition called polycythemia. So in that, the normal, it's not good to have abnormal rise or the abnormal number of RBCs. The drastic change or the increase in the RBCs, that's not a good condition. Though we'll be having the hemoglobin and all, it's not a good condition. Those RBCs of those of no use. So that is the condition polycythemia. Decreasing the number of RBCs is what we call the erythrocytopenia. And the shape of the RBCs, usually in case of the mammals, during the maturation, they lose the nucleus. The RBCs are enucleated biconcave in nature, and the convex and the concave shapes are there. So this is biconcave. They'll be having depression on both the ends, both the sides, and circular. Whereas in case of the camel and the llama, this is oval in shape. And the size of the RBC varies from 7 to 8 micrometer in diameter and 1 to 2 micrometer in thickness. And the color of the RBC varies, it will be usually yellowish in color. And the structure, as I mentioned, during the maturation, they lose their nucleus and the cell organelles. So they don't have any those structures within them. Just the biconcave oval shaped nucleus will be oval shaped cell will be there. And the quality of the hemoglobin in case of the RBCs, as we all know that the hemoglobin is a component, is a globin protein, which is helpful to carry the oxygen. So every hemoglobin particle carries 4 molecules of oxygen during the respiration. So based on that, we can analyze the quantity of the hemoglobin in the RBC. And formation, the RBCs mature, or the formation of the RBCs takes place in the bone marrow. We all have the bone marrow. Erythropoiesis is a process where the RBCs formation occurs. And the lifespan, as I said, since they lose all cells, organelles, and the nucleus, the lifespan of the RBCs is very restricted, like 120 days. After that, there will be a organ, what we call the spleen, which is in association with the liver. So it is popularly known as a graveyard of RBC. After the 120 days of their lifespan, they'll reach the spleen, and there the destruction of the RBCs occur. With respect to the function of RBCs, RBCs take part in the transportation of oxygen and transportation of carbon dioxide. It is mainly involving in the respiratory process. So that's how the transportation of oxygen and carbon dioxide occurs in the RBC, and maintenance of pH. So since it forms the buffer and all, it also maintains the pH of blood. Next, we'll move on to the leukocytes, the white blood corpuscles. The major role of the white blood corpuscles is in the defense, in order to prevent the body from various infections. So the number of the white blood corpuscles varies from 6,000 to 8,000 cubic millimeter of blood. And the shape is usually round shaped or irregular shape will be seen in case of the white blood corpuscles. The size of the white blood corpuscles will be 12 to 20 picometer. Color will be usually colorless. The structures in case of the white blood corpuscles will be having a cell membrane, nucleus, and cytoplasm will be there in their structure. And based on the presence and absence of the granule-like components, we can differentiate white blood corpuscles into 2 types, that is agranulocytes and granulocytes. Agranulocytes are the one, as the name indicates, they'll be having granule-like components in their cytoplasm. Example for them are the lymphocytes and monocytes. Granulocytes are the one which are having granule-like components. Agranulocytes are the one which are not having granule-like components, whereas the granulocytes are the one which are having granule-like components. An example for them are the eosinophils, basophils, and neutrophils. And the formation, obviously, even this divides in the bone marrow, whereas the maturation of the white blood corpuscles occurs in different places. The formation of the white blood corpuscles is what we call the leukocytosis. And the lifespan of the white blood corpuscles varies from 4 to 8 hours in the circulating blood and 4 to 5 days in the tissue. When they are there in the circulating blood, they'll retain or they'll live up to 4 to 8 hours. Whereas when they are there in the tissue, they'll live up to 4 to 5 days. So the next component in the blood is the thrombocytes or the blood platelets. So you all know the importance of the blood platelets. Blood platelets plays a very important role. The count of the blood platelets came into a that was so crucial when it comes to the dengue condition. You all come across the dengue condition, and we all happen to see how the dengue patients have been suffered in the past 2 years. before the arriving of this corona. Dengue was a major one which was infecting the slums and the various regions. And that's how our PHCs also taken a major role in order to prevent the breeding sites of the mosquitoes. So this blood platelets plays a very important role. So when it comes to the number, as you all know that it is 1,50,000 to 3,50,000 cubic millimeter platelets of blood, and the increase in the number of the blood platelets will be represented as thrombocytosis, and the decrease in the number is thrombocytopenia. And the shape of the blood corpuscles, blood platelets will be round or oval disc shaped will be there. And size of the blood platelets will be around 2 to 3 micrometers in diameter. Color of the blood platelets will be colorless. They'll be not having any color. And the structure of the blood platelets usually will be flat, non-nucleated fragments of the cell. And the formation, platelets are also formed in the bone marrow, and that majorly the megakaryocytes. Megakaryocytes are the larger cells of the bone marrow. They'll take up the duty or the responsibility of forming the blood platelets. And lifespan usually they'll stay up around a week, about a week in the circulating fluid. And function, with respect to its function, it majorly involves in forming the clots. When the injury happens, the blood platelets, along with the clotting factors, helpful to prevent or to form a plug, so that there will be the prevention of the blood or the bleeding. It stops the bleeding. With this quick foundation of the blood, the plasma and the blood cells, we'll get into the blood groups. My major topic is the blood groups. So the A, B, O blood group and the RH blood group. See, there are more than 30 antigens are there on the surface of the blood cells, the RBCs, and that leads to different types of blood groups. So consent to my topic, I have to explain ABO blood groups. Along with that, I'm going to explain the RH factor. So why the blood group is very essential? What is the use of the blood grouping? Why we need to know the blood groups? Those are the things we'll be discussing in this consent topic. Well, in this particular picture, I just want to show you that how the agglutination happens within the bloods. Take for example, in the first picture, it is group O, whereas here the dots represents no agglutination, and this clumping like stuff represents the agglutination. Agglutination is nothing but it's a kind of ನಿಮಗೆಲ್ಲ ಗೊತ್ತಿರಬಹುದು, ನಾವು ಮೊಸರನ್ನ ಹೊಡೆದಾಗ ಹಾಲು ಎಷ್ಟು ಕ್ಲಿಯರ್ ಆಗಿರುತ್ತೆ, ಎಷ್ಟು ಫ್ಲೂಯಿಡ್ ಲೈಕ್ ಇರುತ್ತೆ, ಬಟ್ ಅದೇ ಮೊಸರಾದಾಗ ಸ್ವಲ್ಪ ಗಟ್ಟಿ ಗಟ್ಟಿ ಕ್ಲಂಪ್ಸ್ ತರ ಆಗುತ್ತೆ. ಅದನ್ನೇ ನಾವು ಅಗ್ಲುಟಿನೇಷನ್ ಅಂತ ಹೇಳಿ ಹೇಳ್ತೀವಿ. ಸೊ ಜಸ್ಟ್ ಹಾಗೆ ಫ್ಲೂಯಿಡ್ ತರ ಇದ್ರೆ that will be no agglutination will be there in that. So in case of the O blood group, A serum, B serum ಎರಡರಲ್ಲೂ ಕೂಡ agglutination ಇರೋದಿಲ್ಲ, it's clear. In case of the A blood group, agglutination will be seen only in the A serum. And in case of the B blood group, agglutination will be seen in the B serum. Whereas in case of the AB blood group, agglutination will be seen in both the serums. So ಇಲ್ಲಿ ಎರಡರಲ್ಲೂ ಕೂಡ ನಾವು agglutination ನ ನೋಡಬಹುದು. So the ABO blood group was first reported by the Karl Landsteiner in the year 1900, and whereas the AB blood group, he found out the A, B and O blood groups, whereas the AB blood group was later on found out by the Castello and Steny in the year 1902, and they determined the blood groups by the gene I. I represents the isoagglutination, agglutinin. So here we can see it's a chart where we can see the blood groups. As I said, A, B, AB and O blood groups are there, and their genotypes. So if ಇಲ್ಲಿ ನೋಡಬಹುದು ನೀವು ಎರಡು A ಇದ್ರೆ, this is what we call homozygous A blood group. ಒಂದು A ಒಂದು O ಇದ್ರೆ it is heterozygous O group. So Mendelian law prakara it is a codominance. So A blood group individuals ಅಲ್ಲಿ ಎರಡು ತರ genotypes ಇರುತ್ತೆ. Again B blood group ಅಲ್ಲಿ, AB blood group ಅಲ್ಲಿ ಎರಡು ಕೂಡ express ಆಗುತ್ತೆ. That is what I said, codominance, and O is a recessive one. Accordingly, how can we classify these AB and O blood groups based on the presence of antigen and antibodies? As I said, antigen is a substance which is there on the cell membrane, red blood corpuscle cell membrane mele iruvanthadu antigen, and the antibodies are the one which are there in the blood plasma. A blood group individuals will be having A antigen in their red blood corpuscle and antibody B in their plasma. And B blood group individuals will be having antigen B in their red blood corpuscle and antibody A in the plasma. Whereas the AB blood group individual will be having both the antigens in the RBC, but they'll be not having any antibodies. Whereas the O blood group individuals will not be having any antigens, but they'll be having both the antibodies in their blood plasma. And here we can see for whom they can donate the blood and from whom they can get the blood. But usually, see here they have given the possibilities like A blood group individual can donate blood to A and AB. But usually only in the emergency cases, they'll go for this kind of experimentations like or in a very neat condition. But usually they'll go for the specific blood groups like A blood group ಇರುವಂತ ಇಂಡಿವಿಜುವಲ್ಸ್ A blood group ಅವ್ರಿಗೆ ಮಾತ್ರನೇ ಕೊಡಬೇಕು. AB ಗೆ ಕೊಡೋದನ್ನ ಅವರು ಆದಷ್ಟು ಪ್ರಿವೆಂಟ್ ಮಾಡ್ತಾರೆ because they'll be having the antibodies B antibodies that may undergo agglutination with that. So only in the emergency cases ಯಾವುದು ಸಿಗ್ಲಿಲ್ಲ ಅಂತ ಹೇಳಿದಾಗ if we are not with any other options, we'll go for these kinds of things, or else A blood group ಇರೋರು A blood group ಅವ್ರಿಗೆ ಮಾತ್ರನೇ ಕೊಡಬೇಕು, ಹಾಗೆ B ಇರೋರು B blood group ಅವ್ರಿಗೆ ಮಾತ್ರನೇ ಕೊಡಬೇಕಾಗುತ್ತೆ. So next we'll move on to the rhesus factor, RH factor. So this RH factor was named after the rhesus monkey when that was first identified by the rhesus monkey, blood of rhesus monkeys in the year 1940. Again, this was done by Landstedt and Wiener. They found out that particular antigen in the rhesus monkey. That's why they named it as a RH factor, rhesus monkey factor. The rhesus monkey factor or the RH factor plays also an important role during the blood transfusion and also during the incompatibility during the pregnancies. So here there is an image where I can explain the incompatibility during the pregnancy. So if at all, ಒಂದು RH ಫ್ಯಾಕ್ಟರ್ ಪಾಸಿಟಿವ್ ಇರುವಂತಹ ಮತ್ತು RH ಫ್ಯಾಕ್ಟರ್ ನೆಗೆಟಿವ್ ಇರುವಂತಹ ಎರಡು ಕಪಲ್ ಏನಾದ್ರೂ ಮದುವೆ ಆದಾಗ ಅದರಲ್ಲಿ ಮದರ್ RH ಫ್ಯಾಕ್ಟರ್ ನೆಗೆಟಿವ್ ಇದ್ರೆ ತಂದೆದು RH ಫ್ಯಾಕ್ಟರ್ ಪಾಸಿಟಿವ್ ಇರುತ್ತೆ, ಅಂದ್ರೆ ಗಂಡಂದು RH ಫ್ಯಾಕ್ಟರ್ ಪಾಸಿಟಿವ್ ಇರುತ್ತೆ. ಸೊ fetus, if at all the fetus ಗೆ RH factor, since RH factor is a dominant character, fetus ಅಲ್ಲಿ RH factor positive ಇರುವಂತಹ ಎಲ್ಲಾ ಚಾನ್ಸಸ್ ಇರುತ್ತೆ. fetus RH positive ಆದಾಗ it produces the RH factor antigen. So since the mother, there is a direct connection between the fetus and the mother, that is a placenta. ಇಲ್ಲಿ ಫಾರ್ RH antigen, RH positive antigen ಗೆ antibodies produce ಆಗುತ್ತೆ ಮದರ್ ಅಲ್ಲಿ, and that will be those anti-RH antibodies will be remain in the blood stream of a mother after the parturition. parturition ಆದ ಮೇಲೆ ಮೊದಲನೇ ಮಗು ಡೆಲಿವರಿಗೆ ಯಾವುದೇ ಪ್ರಾಬ್ಲಮ್ ಆಗೋದಿಲ್ಲ. after the parturition of the first pregnancy, those anti-RH factor will be there in the mother blood stream. But second pregnancy ಆಗುವಾಗ ಮತ್ತೆ ಮಗು RH positive ಇದ್ರೆ whatever the anti-RH antibodies which are there in the mother serum will fight against the RH. ಇಲ್ಲಿ ಮಗುವಲ್ಲಿರುವಂತಹ RH factor ಮೇಲೆ ಫೈಟ್ ಮಾಡಿ they lead to a condition that is what we call the erythroblastosis fetalis ಅಂತ ಒಂದು ಕಂಡೀಷನ್ ಕ್ರಿಯೇಟ್ ಮಾಡಿ that leads to death of the embryo. So this happens for the second pregnancy. So adrinda if at all RH incompatible idre couples ali, after the first pregnancy there'll be an injection in order to neutralize the anti-RH antibodies that formed in the first pregnancy, so that the second pregnancy will occur without any any complications. Without any complications, second pregnancy kuda aramagi agutte. So what are the importance of blood group study? There's a great value for the blood group study, the blood transfusion. So during the blood transfusion, earlier blood groups ella gotilde idaga, there was so many mishappenings happened. Even many have been dead because of the transfusions occurred. Incompatibility agi tumba jana sat hogidru. when you look into the, or when we study the blood groups, we can actually look out the compatibilities, and we can, if we go for the transfusion, no difficulties will occur in the blood transfusions. And you all know that the importance of blood transfusion these days in the accident cases and in the anemic conditions. Some individuals will go for severe anemic conditions. So in those conditions, blood transfusion is very necessary. So by knowing the blood groups, we can easily go for the safe transfusion. And in order to know certain blood diseases, so by knowing the blood group analysis, by doing the blood group analysis, we can go for studying certain blood diseases and the paternity test. So if there is a dispute between the father, your parental like the disputes idre adana clarify ಮಾಡ್ಲಿಕ್ಕೆ ಕೂಡ will go for the blood group studies. But now we are having the DNA fingerprinting technique. More advanced DNA fingerprinting techniques are there in order to sort out the paternity test and all. And blood group examination is also helpful in the forensic medicines. So the many criminology I mean, the teachers are there, they can make out uh, importance of the forensic or the blood grouping in the forensic. ಎಲ್ಲರೂ murder cases ಅಥವಾ in case of the crime activity ಇರುವ ಕಡೆ ಏನಾದರೂ ಸ್ಯಾಂಪಲ್ ಸಿಕ್ಕಿದ್ರೆ, so they'll analyze the blood groups and all. So in that, helpful in the forensic medicine. And ethnological studies in order to survey or the uh, in order to study the differences in the individuals, ethnological studies have been done. So this blood group analysis is also helpful in that. And anthropological studies in order to see, we can see different kinds of surveys, like even in a recent days, like a blood group individuals are more prone to corona diseases antella helidru. Well, that's all the myth. There is nothing to do with the diseases and the blood groups. So either ಕೆಲವೊಂದೆಲ್ಲ anthropological studies ಮಾಡ್ತಾರೆ. Some blood group individual will be prone to certain diseases, or the certain characteristic features will be similar in those particular individual. So in order to review that, anthropological studies will be done. And even for various experimental purposes, this blood group testing will be done. So this is one of a student is performing the blood group experimentation. This is there in our syllabus, where we'll be getting the antiseras from the um, chemical suppliers. So usually those antiseras will be having the monoclonal antibodies, like the, as I said, the antibody A, B, and Rh antibodies will be provided in that. So the procedure is simple. We'll take a slide. ಮತ್ತೆ ಸ್ಲೈಡ್ಸ್ ಅಲ್ಲಿ ಮೂರು ಮಾರ್ಕಿಂಗ್ ಮಾಡ್ಕೊಂತೀವಿ, ABD ಅಂತ. ಸೊ ಆ ಮೂರು ಮಾರ್ಕಿಂಗ್ಸ್ ಅಲ್ಲಿ ವಿಲ್ ಪ್ಲೇಸ್ ದ ಬ್ಲಡ್, ಒಂದು ಡ್ರಾಪ್ ಬ್ಲಡ್ ನ ಪ್ಲೇಸ್ ಮಾಡಿ ಅಲ್ಲಿ ವಿಲ್ ಅಕಾರ್ಡಿಂಗ್ಲಿ ವಿಲ್ ಆಡ್ ದ ಆಂಟಿಸೆರಾಸ್. ಲೈಕ್ ಎ ಇರೋ ಹತ್ರ ಆಂಟಿಸೆರಾ ಎ ಆಡ್ ಮಾಡ್ತೀವಿ, ಬಿ ಇರೋ ಹತ್ರ ಆಂಟಿಸೆರಾ ಬಿ ಆಡ್ ಮಾಡ್ತೀವಿ, ಅಂಡ್ ಓ ಇರೋ ಹತ್ರ ಆಂಟಿಸೆರಾ ಓ ಆಡ್ ಮಾಡ್ತೇವೆ. ಸಾರಿ, ಡಿ ಆಂಟಿಸೆರಾ ಡಿ ಆಡ್ RH ಆಂಟಿಸೆರಾ ಆಡ್ ಮಾಡ್ತೇವೆ. ಸೊ ಇಲ್ಲಿ ಕ್ಲಂಪಿಂಗ್ ಆಗಿದೆ. ಸೊ ಯು ಕ್ಯಾನ್ ಸಿ ದ ಕ್ಲಂಪಿಂಗ್ ಇನ್ ದ ಬಿ ಅಲ್ಲಿ ಕ್ಲಂಪಿಂಗ್ ಆಗಿರೋದು ನೋಡ್ತೀವಿ. ಅಂಡ್ ಅಲಾಂಗ್ ವಿತ್ ದಟ್ ಓ ಅಲ್ಲೂ ಕೂಡ ಕ್ಲಂಪಿಂಗ್ ಆಗಿತ್ತು. ಇಟ್ಸ್ ನಾಟ್ ದಾಟ್ ವಿಸಿಬಲ್ ಬಿಕಾಸ್ ಕ್ಲಂಪಿಂಗ್ ಚಿಕ್ಕ ಚಿಕ್ಕ ಪಾರ್ಟಿಕಲ್ಸ್ ಆಗಿತ್ತು, ಅದಕ್ಕೋಸ್ಕರ ಅಷ್ಟು ಕ್ಲಿಯರ್ ಆಗಿ ಕಾಣಿಸ್ತಾ ಇಲ್ಲ. ಬಟ್ ಯು ಕ್ಯಾನ್ ಮೇಕ್ ಔಟ್ ಕ್ಲಿಯರ್ ಕ್ಲಂಪಿಂಗ್ಸ್ ಇನ್ ದ ಬಿ. ಸೊ ದ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಆಫ್ ದಿಸ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಸ್ಟೂಡೆಂಟ್ ಇಸ್ ಬಿ ಪಾಸಿಟಿವ್. ಸೊ ಬಿ ಅಲ್ಲಿ ಮತ್ತೆ ಡಿ ಅಲ್ಲಿ ಎರಡರಲ್ಲೂ ಕೂಡ ಕ್ಲಂಪಿಂಗ್ ಆಗಿದೆ. ಸೊ ದಿಸ್ ಇಸ್ ಹೌ ವಿಲ್ ಗೋ ಫಾರ್ ದ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಸ್ಟಡೀಸ್ ಇನ್ ದ UG levels. So these are the references I referred for this particular presentation. Thank you.